I'm ready.
All right, hi, I'm Kirsten Grzybowski, and I'm here with Noah. And together we did, with the guidance of Dr. Lau, we performed a water quality assessment on Connolly's Run above the bridge in Wildwood Park, Radford, Virginia.
Constant monitoring of a stream's health is vital to keeping it healthy. The environment is ever changing, and so is the health of our waterways. Millions of species, including ourselves, rely on these sources of water. Through research, we can learn how a multitude of factors can influence stream health and what can be implemented to improve overall health. During assessments, we check for chemical, physical, and biotic factors as means of reference for quality.
For our stream monitoring program, we did a rapid biology assessment, and we did this by sampling the stream reach. So basically, you just sample one section of the stream, and then you reference it and use it to determine the status of the whole location of like this section of the stream. So we use this type of assessment in order to determine the stream status. So if the stream is impaired or not, and ours was, but we'll talk more about that later. In order to do that, you measure the abiotic and biotic factors, which is things like water chemistry, bugs, and all that. However, the downfall of this type of assessment is that it is quicker, so we can find out if something is wrong, but not necessarily what is wrong, and that's just because it's rapid. So you sacrifice kind of the quality for the timing of it. And we also use it so that we can plan out how to fix the impaired sources and the impaired parts and help determine the restoration methods. So the type of stream, it was very large. In fact, some other students in my class did the same stream. And if you've ever been to Wildwood Park, you know that all of the trails, basically, you can see the stream at some part of it or the whole thing while you're walking or running. There is lots of forested area around it on both sides, on both banks. They're both recreational usage, so both trails as well as the forested areas. The stream sediment is very rocky with the size fluctuating between medium and large rocks. The flow of the stream is majority just riffles, but there's also a decent amount of pools and glides. And the wetted width is about 6 meters, so the distance across. Are you frozen?
We assessed the health of Connolly's Run at Wildwood Park using a variety of methods. We followed the QA/QC training to study our assessment unit, which was the stream reach. We used a Yellow Springs instrument to obtain most of our quantitative data of our stream at Transect A. We took a lot of data with this device. It shows us the temperature, pH, nitrates, salinity, conductivity, and the rest of the data on the slide shown. We used a turbidity tube to find the turbidity of the stream. We assessed different dimensions of the stream's physical habitat at each one of our 11 transects. We measured the longitudinal dimension of the stream in the Thalweg profile. The Thalweg was divided into equidistant sections by dividing our stream reach by 100 and using that variable to take measurements at each point. Our stream reach was 252 meters, so we took a Thalweg measurement every 2.52 meters. We measured the depth in millimeters and classified the channel unit and by either riffle, glide, or pool at each of our Thalweg measurements. We performed a Wallman pebble count method with a metric ruler to quantify the stream substrate size. We used a sample size of 100 pebbles. We used the bobber method to measure surface water velocity by timing how long the bobber took to get from one point to another. We ran 5 trials and averaged the time it took to cover the distance. We used this data to calculate the velocity and area of the stream, which allowed us to calculate discharge as meters cubed per second. The left and right canopy cover was measured using a concave spherical densitometer at each transect. It's pictured on the slide. The percent stream reach canopy cover was calculated by dividing the total number of points measured with canopy cover by the total possible amount of points measured on the densitometer. And we performed a cross-sectional depth profile at each of our transects. We took the wetted width and divided it by 10, and we measured the depth at each of the equidistant points across the width, including the left and right bank. Our team collected a sample of benthic macroinvertebrates at transect A. We used a dip net to collect our sample before we took any measurements and disturbed the habitat. After the sample was collected, we preserved it using ethanol to later classify in the lab. This next section of the presentation goes over the results we found from our stream assessment of Connolly's Run. Table 1 here depicts the microclimate conditions of the day. We collected our stream data. This is part of our QA/QC protocol. You can see it was not very windy day. It was 18 degrees Celsius outside, and it was not raining. Table 2 summarizes a lot of our physical habitat data that we collected. It shows our average wetted width and depth, which were 6.3 meters wide and 151.6 millimeters deep. There's the percent of each channel unit we measured during the Thalweg profile, riffle being our most prominent stream category. The percent canopy cover gives an idea of how dense the foliage is around the bank of our stream. We had a percent canopy cover of 87.4%. From the 5 surface water velocity measurements we took, we calculated the stream was moving at an average velocity of 0.37 meters cubed per second. Turbidity was reading at 120 millimeters the day we collected data. We were able to see through our sample to the bottom of the turbidity tube. And our rapid habitat assessment score, which I will discuss shortly. Table 3 shows the water chemistry data that we collected from transect A of our stream reach. Water temperature was around 16 degrees Celsius. We measured a pH of 8.32, which is starting to lean closer to basic than neutral. We took measurements of dissolved oxygen, conductivity, total dissolved solids, salinity, and nitrates. We noticed our nitrates were pretty high as well and our conductivity. Table 4 summarizes the rapid habitat assessment we performed on Connolly's Run. This is a quick evaluation of the habitat quality, which is essential to our assessment because habitat quality and biological diversity go hand in hand. Connolly's Run scored proficiently on only a few of the metrics that this assessment tests for, such as channel flow status, right bank stability, left and right bank vegetative protection. Overall, our stream scored an overall of 109 out of 200 points, which means it is impaired because of this assessment, based off of this assessment. Table 5 is the distribution of insect species we collected from our sample from Connolly's Run. Our sample was preserved with ethanol, then sorted through under a microscope for 30 minutes to keep consistent data amongst the teams. We found 14 different insect families and 182 insects in total. The 2 most prominent insect species we found in our sample were the net spinning caddisfly and the fingernet caddisfly. And the final table here summarizes our Virginia Stream Condition Index, or our VCSI score. This index uses metrics to quantify insect abundance as well as the distribution of specific indicator species into a single numerical score. The VCSI score our team calculated for Connolly's Run is 60.5, which deems it not impaired.
So going off of what Noah was talking about, of what was impaired, he talked about our dissolved solids were definitely high and our nitrate was definitely high. We're categorized as number 4, which means that at least 2 out of our 7 parameters are fully supporting aquatic life. We actually had 4, but in order to be a category 2, you have to be between 5 and 6. parameters that are fully supporting aquatic life, so that's why we're number 4. Um, our dissolved solids and nitrate impaired is probably because there's obviously many people that go around the streams, so there's a lot of traffic, which means that there's a lot of pollution, so that's always a factor. It could also be high because before our assessment there was heavy rainfall, making the variables change, and also just stirring up the floor of the stream and making it mucky and such, and just bringing everything up. It's also could be high because the banks on the stream, the withering of it, and the runoff, especially if they salt the trail paths. But also the runoff from the fertilizer affects the stream, and the banks bring that in as well. So it changes the variables. So a lot of these impacts aren't really avoidable, like the weather and the fertilizers are pretty much always going to be used. So the best way to just keep it healthy is keep monitoring the stream and maintaining what's already there, like organized stream cleanups and just the park being open from only dusk to dawn. And then we just wanted to thank everyone. So we wanted to thank the City of Radford and Pathways for Radford for creating and maintaining Wildwood Park, Radford University for providing us with the opportunity to conduct a stream assessment, and also for providing all the equipment, the Office of Undergraduate Research and Scholarship for providing a space for the team to present our work, the Mayor of Radford and City of Radford for letting our team use the park, and finally Dr. Lau and our TAs for helping us along the way, especially Danya for helping us out in the field. Here's our literature cited. Any questions?